I'm Mrs. Sloan, and we are moving on to part 2 of chapter 6. So chapter 6 is all about metabolism, energy, and enzymes. So we've already talked about energy, so now we're going to start talking about enzymes. All right, so I'm going to jump right here to metabolic pathways. The second I want to present here. Oh, look, I'm in the middle again. Always does that. Okay, so, and get a pointer. Okay, so on your notes, and remember group shared notes are down at the bottom, and you fill out the one on the left, on the column on the left, and add pictures on the right, okay? So metabolic pathways and enzymes. So this kind of looks confusing, right? This is a series of metabolic pathways. Maybe you can find some that you're familiar with. Look right here. Carbon fixation. That sounds a little bit like photosynthesis. I can see NADPH and ATP. Look at this fatty acid synthesis. I'm looking over here at the citric acid cycle. Another name for that is the Krebs cycle. So these are all a series of metabolic reactions, an orderly sequence. So put that on your metabolic pathways, that this is an orderly sequence of linked reactions. An orderly sequence of linked reaction, and each step is catalyzed by, name it, a specific enzyme. Each one of these steps is catalyzed by a specific enzyme. So if we think about energy, right? Let me move myself, okay? There's a lot of energy in bonds that you eat. So let's say right now you're like, you know what? I want a donut. And so you go and you get a donut and you eat that donut. There is so much energy in all of those bonds of all those all the carbon and the hydrogen, right? If you could break and release all of that energy at once, you would like spontaneously combust into flames, right? It would release so much energy. So what happens when we try to harvest the energy, the potential energy in bonds from the food we eat, we release a little bit at a time. Just a little bit in small increments so we can capture that, right? If you're trying to drink out of a fire hose, it's going to rip your face off, right? But if you're drinking out of a hose in your yard, you can manage that amount. You can manage that energy and transfer it to the other bonds that you want, that you would be useful. Okay, so metabolic energy is captured more easily if it is released in small increments. All right, so next what I want to talk to you about is catalysts. So catalysts, there's a part of their surface that will interact with the reactant so you can facilitate the steps of a reaction. And I'll show you this in class because this is an animation that I don't have here. Okay, so let's look right here. Let's look at the surface of this catalyst. This is an enzyme, right? So on this enzyme, side note, enzymes are made out of proteins, and so we just want to remember for just a quick minute, right, about the structure of proteins, right? There's primary, secondary, tertiary, and quaternary structure. So the primary structure is the what? It's the sequence of amino acids and the number of chains you have. The secondary structure is either an alpha helix or a beta pleated sheet, and that is not due to the R groups, right? It is just due to the N terminus and the C terminus end and the way they are attracted to each other in a chain of amino acids. The tertiary structure is dependent on the R groups, right? Because you have your hydrophilic, hydrophobic interactions, and that's the folding pattern imposed upon the secondary structure. And then if you have a quaternary structure, that's if you have more than one chain. So this enzyme, when you see it, is just this yellow on this slide. Remember, it's really made out of amino acids. It's in a special conformational shape. If it doesn't have its right shape, it can't do its job, all right? Structure equals function. So here, this portion of the enzyme right here is referred to as the active site because that's the part of the enzyme that will interact with the substrate directly. It will bind on. Okay, then when you have those joined together, this is an enzyme-substrate complex. Remember, you are having these two molecules interact with each other, so bonds will change as a result of that, right? And then as those bonds change, then you're going to the enzyme releases its now products. So would this be an anabolic reaction or a catabolic reaction? Are we breaking or are we building? hopefully you're going to recognize that this was a catabolic reaction because you broke something down. Look at your product with your two parts here, right? Okay, so on your notes, what I want you to have is that catalysts are substances that can change the rate of a reaction without being altered in the process. Without being altered in the process. So there may be a temporary conformational shape right here that changes, but notice it bounces right back to its regular shape, okay? Without being altered in the process. And it's changing the free energy. Remember, free energy is energy available to do work, right? Changing the free energy change, and you have a difference in your reaction, difference of the reaction. So we'll talk about that, okay? Look at here, okay? So if you look at this, the whole thing, right? Now you can see all the individual amino acids that are making up this protein. And if you think of this as like a mouth, the mouth is the active site. So when it binds with the substrate, do you see how it looks like its lips are closing? So that is a temporary change in its shape, just as it's bound to the substrate. Okay, and so we will talk about that later in the notes. It's called, if you skim down in there, it's the induced fit model. Okay, so we're going to come to that in just a minute, but I wanted to show it to you there. Okay, so let's talk about what are enzymes. What would an enzyme be? So we call them organic catalysts. They would be organic, right? Because they're built out of carbon, right? Complex carbons, organic catalysts, and they are usually a protein. They don't have to be a protein, but they are usually a protein. And it can increase the reaction rate by more than, ready for this, 10 million times faster. So what are the odds of this reaction that we just saw right here occurring, right? With or without the enzyme, it's 10 million times more likely with the enzyme than without. So you can see that's why you need an enzyme, all right? And there are other than proteins, things like ribozymes. Now, what do you hear in ribozymes? Do you hear ribosome, right? Ribozymes are made out of RNA, and they are involved in the synthesis of RNA and also proteins on the ribosome. So it's the making of the RNA and also proteins on ribosomes. The substrate, by definition, is the reactant. So that's what you're seeing right here in its mouth. That would be like the substrate, all right? Whatever is binding, whatever the enzyme is working on. All right, let's take a look at this picture. I'm going to make myself smaller here. So there I am, okay? The enzyme's slight change in shape is what's facilitating this reaction. So here's your substrate, the part where it's binding, that's the active site, right? That's the active site. And then it's reorganizing the bonds in the substrate at critical areas. And so then your product is here, okay? But again, it maintains, it can be used again and again and again. So on your notes, what I want you to have is active site is the part of the enzyme that binds temporarily with the substrate. It just binds temporarily with the substrate. And we'll get into more details about what can influence that, but for right now, what can influence the active site? Any factor that alters the active site, like temperature or pH or an inhibitor, can change the shape of the enzyme, anything that's going to change the shape of the enzyme and affect somehow its active site, right? Change the shape of the enzyme and it'll change its functionality, So enzymes can become denatured, just like we learned about proteins becoming denatured. It can change its allosteric configuration in space, which then affects its functionality. Okay, so the induced fit model, I gave you everything for that. That slight change it goes through. And then the next part on your notes is that enzymes are not consumed in the process, right? An enzyme does its job, releases its product, does its job, releases its product over and over and over again. And we will talk about the rate at which a reaction occurs. We will talk about the factors that affect the rate of the reaction. But because they are not consumed, you only need a very small amount of them. So enzymes are not consumed by the reaction, so only a small amount is required. And then we already learned about enzyme naming. I taught you that a couple chapters ago, right? So they're named after their substrate, right? So we learned about sucrose, right? And to digest sucrose down, you need the enzyme sucrase, and we learned that it breaks it down into glucose and fructose, right? So I gave you all of that already in the name. All right, now let's think back to part 1 of this chapter when we talked about exergonic and endergonic reactions, right? We talked about spontaneous reactions that could occur, but there's always a little hurdle, right? Because you have to invest a little energy. So if you look at A versus B, right, who has more free energy, A or B? A does, right? So this is representing that A has more energy, free energy than B. Remember, energy is not created or destroyed. That's thermodynamics number 1. But every time it changes forms, right, we lose some as heat. What I am talking about right here is the energy in the reactant. This reactant A has more potential energy than the product, okay? So we're going to be releasing some energy here, right? Our delta G is going to be negative. We'll hopefully harvest some of that energy, couple it with a reaction that is endergonic so you don't lose all of it, okay? Now, but take a look right here. There is a hurdle right here. This hurdle may get in the way of this spontaneous reaction from occurring if they can't push that ball or that rock up over that hill. So take a look at this picture right here, okay? Once again, A and B, and here's C and D. So our delta G would just be measured from this point to this point. This is our delta G, and it's going to be negative because we lost energy. But this hurdle of right there, it makes sense. It's called the energy of activation. It's the hurdle that you have to get over in order to break that down. Now, if you have a fireplace, I don't know if you have a fireplace in your house, and with all the fires going on right now, maybe this is not a great analogy, and I pre-apologize for this, okay? But if you think about, let me just get bigger here for a minute. If you were going to light a fire in your fireplace, right? You might put some kindling in there, maybe a little bit of paper, and you'd put some logs in there. There's a lot of potential energy in those logs, right? But it just doesn't burst into flames in your fireplace. It does not burst into flames. You have to get the party started, right? You have to invest a little bit of energy, and then once you get that ball rolling, then the fire will take off. So you use a match or something, and you get a little bit of the paper burning, and that causes a little bit of the kindling to burn, and that kindling starts to get the log to burn, and all you needed was that one little match, and everything then starts burning. We saw this here in Southern California, right? Somebody was celebrating with some pyrotechnics, and that one little thing has destroyed thousands of acres here in Southern California because it just started releasing energy, which facilitated the next reaction and facilitated the next reaction. So that is called the energy of activation. All right, so on your notes, you have energy of activation. It's defined as E sub A, and it's the energy that must be added to cause the molecules to react, to get to C and D here. The energy that must be added to cause the molecules to react. Now, the benefit of having this right here, this little hurdle, is that you don't fall apart all the time, right? The hurdle of energy prevents molecules from just spontaneously degrading, So let's compare with an enzyme and without an enzyme, our energy of activation, okay? So here's our starting point. It's just like one block up here, right on this ridge. And then here's the ending point, just one block down. But notice, look at this hurdle, the energy of activation, right? So you have a lot of potential energy, but you can't access that potential energy because that hurdle right there is so large. What an enzyme does is it lowers, it makes the energy of activation lower. It is not changing your starting point. That's key that you see that. It is not changing the energy of your ending point. Your delta G remains the same. The only thing that you're changing is your ability to start that reaction going. That's the energy of activation, and that's what enzymes do, is they lower that hurdle. Okay. Now, one thing that could make a lot of reactions go, right, is just heat. Heat can make a lot of reactions go forward. But the problem is there's no control in that. You're not just making this specific reaction go or this specific reaction. You will fire up all of the reactions. So enzymes are amazing because they lower the hurdle of a specific reaction that you need to occur. Okay, so enzymes speed up reactions by lowering the inherent E sub A, energy of activation. They don't change the final energy content. They don't change the final energy content. So let's look here. I think I have another picture of that for you, right? So here is the energy of the reactant. Here is the energy of the product. Look at these two lines. The purple one is without an uncatalyzed reaction. You don't have a catalyst. Our delta G is still negative, right? But here is with an enzyme, right? With an enzyme, we're lowering the hurdle and it makes that reaction more likely to go forward. Okay, so next, what enzymes you have are going to determine what reactions go forward, right? If you don't have an active enzyme, it's not going to do its job, right? So on the different types of reactions that we have, let me just go through a few slides. There's not notes on this. This is just a little review. This would be a synthetic reaction. Another reaction, another name you could think of for this is you're building. So this is an anabolic reaction, whereas this one would be a catabolic reaction. Okay, I have some other ones here you might have reviewed in chemistry. Okay, the different types of reactions, right? Synthesis, decomposition, single replacement, double replacement, right? These are all reactions that are going to be facilitated by an enzyme. All right, now, enzymes can alter a product. In this case, you're seeing corn is converted to high fructose corn syrup, which I do not recommend for you, by adding enzymes. And so because of that, then this is a biological novelty for our body. Our body doesn't work well with this high fructose corn syrup, but we'll save that discussion for another day. All right, so factors that affect enzyme activity. Factors that affect enzyme activity. So we'll go through a couple of them here. So just take a minute to look at this graph, see if it kind of makes sense to you. So here is a substrate concentration. It's increasing as we move along our x-axis here from left to right, right? And here is our rate of our reaction. When we have very little substrate, right? When we have very little substrate, then you can facilitate that rate of the reaction is going to be very, very quick. But as you start to increase how much substrate you have, you only have so many available active sites, so it's going to start to slow it down. Notice what this is right here, saturation of active sites. If all your enzymes active sites are full, you can see that that would slow your reaction rate down. So substrate concentration, increase substrate or enzymes, and you will increase the reaction rate, right? If instead of increasing the substrate, I increased the enzyme number, right? So we could do either one of those to regulate. Right here, this is that inflection point, the point of saturation, where it's not going to affect the reaction rate any further because all of our enzyme active sites are full. All right, let's take a look at this graph. Okay, these happen to be both enzymes that break down proteins, pepsin and trypsin. Okay, pepsin is found in your stomach, and trypsin and chymotrypsin are created by your pancreas and secreted in your small intestine. Notice the pH and what they work best at, right? Pepsin in your stomach has a very low pH. In fact, it is an inactive precursor as pepsinogen, which gets activated by your pyloric gland secreting hydrochloric acid, which converts it to an active form of pepsin. So you just don't digest your stomach, right? Otherwise, how would you store pepsin? Because your stomach is made out of protein, right? So it gets activated by a very low pH. You have mucus lining your stomach to protect your stomach lining, unless you get an ulcer. That's another story, another day. All right, so this is where pepsin works, and then here is where trypsin works. So that is going to affect, the pH will affect, here trypsin is in your small intestine, the activity of an enzyme. So I gave you that optimal pH, where the enzyme maintains its structural configuration and therefore its active site. right? Because as you change the amount of hydrogen ions available, that's going to affect that tertiary and quaternary structure that we talked about earlier. Okay, here's temperature. So enzymes work at a specific temperature. We've talked about proteins before and denaturing proteins, right? And how you can do that permanently. And so this temperature right here is where this particular enzyme works best. So temperature is typically, as the temperature rises, you're going to facilitate any reaction because there's more kinetic energy in the system. The enzyme activity rises up to a point, and at this inflection point right here, you have denatured that enzyme and it's going to lose its functionality. Okay, next. Okay, when you look at the way certain organisms work, like ectothermic organisms, right? Because they cannot maintain their body temperature, then their activity as an organism is going to fluctuate depending on the temperature of their environment, right? Because they need their enzymes to be at the correct temperature in order to function. So they would be sluggish in the morning. Like if you were going to go for a run in the hills behind my house, right? I would rather go on the run in the hills in the morning than in the afternoon because I'm always afraid of rattlesnakes and we see them all the time. And you have more activity of rattlesnakes in the heat of the day because their enzymes are at the right temperature than you would in the morning. All right. Whereas if you look at this polar bear, right, if you run into this polar bear on your run, he's going to wipe your face off no matter what the temperature is out because he's an endotherm and he can maintain his body temperature. All right. Next, we have, some Siamese cats. Now, these cats have inherited a mutation that causes an enzyme only to be active at a cooler temperature. So part of their body where it's cooler is one temperature. And then in other places where it might be warmer, then you have some different enzymes that are activated. And that's why you have different colors of Siamese cats. Okay. Now, sometimes enzymes need something to help it, to get it going, to kind of flip a switch, okay, to activate that enzyme. And that would be a cofactor. So sometimes it binds at a secondary site over here, this cofactor, an inorganic cofactor, meaning it's not a carbon-based substance, like maybe it's a vitamin, right? And so then it will then make it so that it's in the right conformational shape to bind to that substrate, or maybe it's right here in the active site. So enzyme cofactors and coenzymes. Cofactors are an inorganic ion, for example, like zinc or iron, okay, an inorganic ion. Coenzymes are still non-proteins, okay, coenzymes, but they are organic. So I misspoke just a second ago, sorry. I think I said vitamins on the slide before, forgive, sorry about that. Coenzymes are small molecules tightly associated with an enzyme that participates in the reaction, often by kind of binding with the substrate as well. So in this case, when you have vitamins, and that's why you need vitamins in small amounts because your enzymes can be used again and again, if you don't have that vitamin, then it will not activate that enzyme to get the product that your body needs. So on coenzymes, a non-protein organic molecule, we're going to learn about NAD in cellular respiration and NADP. Those would be examples of coenzymes. And vitamins are often a component of coenzyme. I think I gave you everything for that part. All right, another way you can add this to your notes as well on factors affecting enzyme activity is phosphorylation. Now in phosphorylation, I think you're probably thinking like ATP, right? Adenosine triphosphate, a phosphate group. We've looked at phosphate groups before. So here you have this inactive protein, but a kinase is adding phosphate, ACE like enzyme, right? Is adding a phosphate group, which is then activating this enzyme so that it can do its job. And we'll see a couple of different examples with that as well. Okay, so now we're going to talk about some other ways to regulate enzymes. So we've already talked about substrate concentration, also enzyme concentration is going to regulate like how much product you get. We talked about pH, we talked about temperature, we talked about cofactors, and we talked about coenzymes, right? All of those things can affect enzyme activity. The last part on here is about enzyme inhibition. How do you turn an enzyme off, all right? Now, there are two categories of inhibitors, a competitive inhibitor and a non-competitive inhibitor. If you notice, they both have the word still inhibitor, so they're still inhibiting either way. But what a competitive inhibitor does is look what it's doing right here. It's binding to the active site. It's competing, that's why it's called a competitive inhibitor. It's competing with the substrate for the active site. Whereas in a non-competitive inhibitor, it's binding to a secondary allosteric site over here, right? And notice though what's happening to the enzyme. Because this other region has bound onto the enzyme. Remember, tertiary and quaternary structure, it's causing a rearrangement of the bonds within, right? It could be like hydrogen bonds, etc., right? Within that enzyme, because it's a protein, so it's changing its active site and turning that enzyme off, and you are no longer going to be able to bind to that substrate. So enzyme inhibition occurs when a molecule, the inhibitor, binds to the enzyme and decreases its activity. Okay, so on non-competitive inhibitor, what you're seeing right here, a non-competitive inhibitor binds to the enzyme at a location other than the active site, And the second site is called the allosteric site, which, if bound to the inhibitor, directly changes the shape of the active site, Now, I don't want you in your brain to go, oh my gosh, you're turning off the enzyme. How mean, okay? You do not want all enzymes to be running 100% all the time, right? We want to have it be regulated so we can maintain our homeostasis. So this is a normal feedback mechanism in cells. So let's look at this one specifically. This is in product inhibition. So I want you to look at this metabolic pathway, okay? A to B, B to C, C to D, D to E, E to F, which is your end product. Each one of these reactions in this metabolic pathway needs a specific enzyme. So to go from A to B, you need enzyme 1. B to C, you need enzyme 2, right? You can see that. Now, this first reactant, A, binds right here to the active site of enzyme 1. And then this A becomes then B, okay? Now, if you look right here, this end product, ultimately, if you've done this a bunch, you start to build up that product, which is F. F, notice it can bind to this allosteric site, the secondary site here. And when it binds, look at how the active site has changed from this curve right here, right, to this corners right here. So now the first reactant A can no longer bind to the to that active site. This shuts down the whole metabolic pathway. And look at how efficient this is. You don't have to shut down each enzyme in the pathway. You only have to shut down the first one. Because if you shut down enzyme 1, you don't have to worry about shutting down the rest of the enzymes because there will be no B or C or D or E. So end product inhibition is a form of negative feedback, like of self-correction, okay? Negative feedback. The final product inhibits the initial reaction of the metabolic pathway from occurring. It increases the efficiency of the pathway by turning it off when the end product accumulates in the cell. That was easy, right? Okay, and then lots of different things can affect. I don't want to just make it as simple like a cartoon, but lots of different things can affect. And we can see pH and where the inhibitor is, or if there's some other substance that's also bound to it. Okay, and I think you have everything for competitive inhibition that we talked about before. Remember, competitive inhibition is you're binding to the active site. Both the inhibitor and the substrate compete for the active site. Okay, the last thing we have to talk about in chapter 6, which I will put in a very short part 3 video, is we're going to talk about oxidation and reduction reactions and metabolism, because that's going to set us up for the next 2 chapters, photosynthesis and cellular respiration. Okay, that's it.